Welcome, everyone, and thank you for joining us tonight to learn about the exciting progress being made in colorectal cancer research. In 2022, the GI Research Foundation launched CA Cure under the leadership of Dr. David Rubin, our scientific advisor. I quickly learned as a patient advocate that the standard of care for many colorectal cancer patients didn't offer enough options for the families, for the patients going through the disease. So in my role as the chair of the GI Research Foundation, I embarked on extensive research and talked to many, many prominent doctors. We worked to identify potential research projects that would be important and interesting to improve the standard of care by driving advances in immunotherapy, personalized vaccines, and other really important research. Working closely with Dr. Rubin and a team of scientific advisors from across the country, the GI Research Foundation created a quick and nimble funding program that has since put over $20 million in the hands of scientists and doctors. 11 leading investigators have been funded at 10 institutions across the country. In tonight's session, you'll hear about a few projects that are advancing the diagnosis and treatment delivery mechanisms for colorectal cancer. We have identified some of the best minds in the country, and their progress is really impressive. We're thrilled to share it with you. The CA Cure funding program has reinforced my commitment to the GI Research Foundation. With our unique and long-standing collaboration with the University of Chicago Digestive Diseases Center, we rapidly deployed donor dollars for maximum impact on patients' lives. We make our research investments using a robust scientific review process that ensures effective use of those dollars without too much red tape. We're committed to being innovative and responsive and providing early dollars to bold ideas. These have become the most meaningful projects currently in the digestive diseases space. What you will hear tonight is undeniable proof that science is powerful. I have a lot of hope because of the progress of the research we funded. On March 20th, we will have a second session with additional projects. That session will focus on exciting clinical trials that are in testing now. So please look for an email invitation to the session, and we hope you can join us. Now I'd like to introduce Dr. David Rubin, Chief of the Section of Gastroenterology, Hepatology, and Nutrition at UChicago Medicine. As the Foundation's scientific advisor, he is instrumental first in helping us launch CA Cure, and then to help us through the rigorous scientific review progress. Please join me in welcoming Dr. David Rubin.
Thanks, Katie. And I want to welcome everybody. As you heard, I am a professor at the University of Chicago, but I have the distinct privilege and pleasure of being the chief scientific advisor to this GI Research Foundation. For those who are new to the organization or just logging in tonight and learning more about it, I did want to just point out that this is an organization that was created by grateful patients and friends of a legend in GI named Joseph Kersner in 1962. And their chief mission at the time was to support what he thought were important priorities in the field. And they gave him broad latitude to identify those priorities, and then they went to work to raise the money so that they could address them. Over the many years that this organization has existed, I will highlight to you that there have been groundbreaking achievements in science and incredible support of young investigators who went on to have illustrious careers in science and in medicine, myself included. Not that it's necessarily as illustrious, but I will certainly acknowledge that as a fellow, I was funded by the GI Research Foundation, and I owe part of my career to them. Now, having said that, the origins of the organization were to support the section of gastroenterology, but when I became chief of this wonderful section, it was clear to me that we should broaden not just the mission of what we were doing at the University of Chicago to include a broader approach to digestive diseases, but also that we should change the bylaws of the GI Research Foundation to incorporate digestive diseases broadly as well. And I'm very pleased that our board adopted that strategy in 2014, and we continued to grow over the subsequent years. As you already heard, there became an urgent priority to address the unmet needs of colorectal cancer, and that priority came to our attention in part because of a dear board member and friend and others who have suffered from this difficult disease. I will tell you personally that it also rang true to me because it was my grandfather, Grandpa Phil, who died from colon cancer when I was in college. And I have a personal connection to that particular disease that has affected me and in many ways led me to choose a career in gastrointestinal diseases and for me to do what I do. So when we needed to change or address the important and pressing needs and urgent priorities of colorectal cancer, it was more than my pleasure to do this on behalf of our board members and specifically Katie and her husband Leo, but also I felt that it was my absolute responsibility to do so. So the CA Cure initiative was created out of this to address these urgent needs, to work to identify scientists who are on the cutting edge, and to break with tradition that the GI Research Foundation had long had built in, which was to fund people who were at the University of Chicago, but rather to identify people who were either in collaboration with us or who shared the same vision and missions as what we were trying to accomplish in the GI Research Foundation. In order to do that, we created a new review process that had to meet the needs that Katie set, which were that it had to be urgent, it had to be fast, and it needed to be mobile and happen as quickly and as soon as possible. Thankfully, our board was on board with this, and we created an entirely new peer-reviewed scientific process and then let appropriate folks apply for it. And of course, I do want to acknowledge very carefully and specifically that it was Katie Chudnovsky and her tireless research that identified some of the people you're going to hear from today and who continues to lead our organization by pushing the frontiers of what we're trying to do, not just as a board and as a foundation, but really we should all acknowledge and credit her for advancing the care and the treatment and ultimately the cures and preventions of colorectal cancer. So for that, Katie, I want to thank you. And I, of course, want to thank Leo, your husband, in your efforts to do this and your incredible generosity. And with that, I'm going to step back now and let us hear from our incredible scientists who are going to share some of the advances they're doing, but not without saying one last thing. If you're logged in today, or if you're watching a recording of this and you are so moved, I want to make it clear to you that you can make a difference. Every gift to the GI Research Foundation helps us in this mission, and every individual, Katie as the shining example, can change the world, and we want you to be part of that. So please join us and support this work. Thank you so much.
Thank you, Dr. Rubin, and thanks for being our tireless leader. So tonight, our first presenter is Dr. Ali Salem from the University of Iowa. Dr. Salem leads an amazing lab and many brilliant scientists in Iowa. As he will explain, colorectal cancer often presents with rare multiple mutations that are resistant to standard treatment. Effective treatment options require multi-pronged approaches. His work focuses on improving the delivery method of these innovative treatments using small targeted delivery vehicles. Please join me in welcoming Dr. Ali Salem.
Thank you so much for the introduction, Katie, and I'm looking forward to giving an update on the research that we've been doing, and that was a really great introduction. So what I'm going to do is I'm going to share some slides that I have to sort of give an update of the work that we've done. So I'll just go through each of the steps of what we've done, and then we will welcome any questions at the end of this presentation. I really want to thank the Gastrointestinal Research Foundation and the leadership shown by Katie and Leo for supporting this research efforts. And we're excited to show some of the progress that we've made on this project. The focus is on development and optimization of targeted drug formulations for colon cancer. The team that is helping on this project is myself, Dr. Sean Geary, who is an immunologist, and then Krishna Patel, Dr. Lokesh Jadadam, Dr. Divya Bhatt, Dr. Ramkrishna Sen, and Dr. Muhammad Al-Nator. They represent a wide range of disciplines from formulation scientists, pharmaceutical scientists, cancer biologists, immunologists, all working together to sort of move this project forward. In terms of the progress that we've made, I just want to quickly briefly describe our goals in this project. So the main aims of this project are to develop a multi-pronged therapeutic approach to colorectal cancer using small molecule inhibitors and chemotherapeutics delivered in soluble form or in nanoformulations. And we have a laser focus on one type of rare colon cancer, which is a BRAF V600E p53 mutated BRCA1 mutated colorectal cancer. And for that reason, we're doing a lot of work to develop the resources to understand how this cancer works, how these mutations work together. And it's very important to do the development of the cell lines with these mutations because they don't really exist. And in all of the efforts that we have gone to identify suppliers or banks or other places, we just couldn't find these particular combinations of this rare combinations of mutations. And so we've been developing cell lines with these specific mutational combinations. And then we're assessing ability of soluble drugs to affect CRC growth viability in vitro and in vivo. And then from there, we're generating and characterizing a suite of nanoformulations, encapsulating the target drugs of interest that can really effectively target these particular combinations of mutations. And then the final step is to test it in vitro and then in vivo for treating colorectal cancer. Now, the reason why this is so important is when you have a drug or a combination of drugs that might work with one type of colorectal cancer, if you have different gene mutations, that combination may not necessarily work as effectively with a specific combination of gene mutations. And so creating the specific disease, the rare disease, and then trying to create formulations that target it will be the most effective way of finding an effective and efficacious combination. So the approach that we've been using to generate the cell lines with the BRAF V600E, the p53 mutations, and the BRCA1 is using CRISPR-Cas9 technology. And what we've done is we've got both mouse cell lines and human cell lines that we've been generating using the CRISPR-Cas9 approach. We've done HT29 and COLO205 with the BRCA1 knocked out, and then CD26 with the BRCA1 knocked out, and the p53 R245W, which is equivalent to a human p53 that we're particularly interested in, and then BRAF V637E, again, with a target sequence that we're interested in. We've done HT29 human cells, COLA205 human cells, and then CD26 mouse cells to try and get these three oncogenes all present. And in HT29, we already have two present, and we just have to introduce the BRCA1. With the COLA205, we have two of them, and we need to introduce the BRCA1. And with the CT26, it was all three. So CRISPR-Cas9 has been the approach that we've been pursuing for that. Where we stand in terms of the preparation of these p53, BRAF, BRCA1 specific cell lines, in the HT29, we have clones generated. We're doing quality control testing, and expansion of these cells are in progress. For the COLA205, we have clones generated where we're doing the BRCA1 deletion. Quality control testing and expansion of these cell lines are in progress. And then for us to be able to do a lot of our development work, we have a CT26, which is in the mouse model, a mouse cell line. We have p53, clones have been generated, QT testing and expansion of the clones with p53, R245W, and BRAF V637E. And just to point out that this particular sequence is the mouse equivalent of the human V600, which is why we've chosen it. So we've tried to look at the most appropriate homology between those sequences. And we've got then the BRCA1 that's partially deleted. And this partial deletion means that we're doing further optimization and CRISPR-Cas9 delivery to generate a fully BRCA1 knockout along with the BRAF and p53 mutations. Once we get this fully completed, it's going to really be like an incredibly valuable tool for this rare cancer. In terms of work on goal 2, we've been evaluating paclitaxel, small molecule inhibitors, in particular BRAF V600E and encorafenib, vermafenib. And then we've been looking at MEK inhibitors, trametinib, which can overcome resistance to BRAF inhibitors, PARP inhibitors, in particular olaparib, rucaparib, and talazoparib, and then a VEGFR inhibitor, nintedanib, which is ostensibly used as an anti-VEGF agent, but at low concentrations can also be used for specific mutations in cancer, as we published in Nature Nanotechnology a number of years ago. So in the in vitro cell viability assay results, we've got concentrations of drug at which 50% die, so that's the IC50, and we're looking at values lower than the IC50 to look at the drug potency. And the order of the IC50 that we've seen so far is trametinib, followed by paclitaxel, encorafenib, vermafenib, rucaparib, talazoparib, nintedanib, and olaparib. So we have an order of potency in terms of the overall drugs that we've been screening. And again, all of these in COLA 205, HT29, CT26. We've also looked at synergy results, and we've looked at combination index results to see which ones are additive, which ones are synergistic, what are not worth pursuing. And drugs that we've identified have clear potential include paclitaxel, trametinib, nintedanib, and encorafenib. Drugs that we have not continued with so far is vermafenib because of the IC50 being too high. And so we do have some target drug combinations that we're seeing are worth pursuing for synergy between them. And then in goal 2, we're looking at combinations of 2, 3, or 4 of the drugs in soluble form, in particular paclitaxel, encorafenib, trametinib, and nintedanib, and looking at them in a therapeutic CRC HT29 animal model. And again, with P53 mutation, BRAF V600E, and a BRCA1 component to it, so that we really have this combination of genes that we're looking at to see whether this combination works effectively. And so you can see the steps. We induce the tumor. This is a colorectal cancer tumor. We treat with the combinations. We monitor tumor size. We look at toxicity, sort of standard things for looking at tumor regression and mouse survival. In terms of the combinations of soluble drugs that we've identified so far, no treatment, you can see it's an appropriate control working really well. The colorectal cancer really does take off. We see the decreases that are most promising are trametinib, encorafenib, paclitaxel, and nintedanib, but there's also a significant change in weight, meaning that toxicity is something that we need to be monitoring. And we see that trametinib, encorafenib, and nintedanib are producing equivalent results, but without that same degree of toxicity, and indicating that these are both combinations that we should pursue further. And we do see some distinct differences between the combinations. So you've got no treatment, and then trametinib, encorafenib, and nintedanib is highlighted, and the next one is also highlighted. And you can see the results on the side showing the difference in no treatment versus the treatment of the combination of drugs and how much that regresses. So the next thing that we want to do is that we know that various nano formulations can give you greater accumulation in the tumor and reduce accumulation in non-target cells, thereby reducing adverse side effects and increasing efficacy. And so we've selected two nano formulations that are made out of materials that have an FDA record of safety and approval. PLGA has a track record in the clinic through a variety of formulations, and then the liposomal formulations have been widely used in the mRNA vaccines and also in doxorubicin as a Doxil. So these are agents that are well safe, with a strong track record of use. And we've loaded them. You can see the SEM and TEM, they're spherical and smooth in appearance. We've done particle size characterization, encapsulation efficiency, looking at ratios. And you can see the schematics of what these groups look like. And we've done it with encorafenib, trametinib, paclitaxel, and nintedanib. And we've even tried co-loading agents as well. So where we're at right now is that we are in progress for making the specific mutated cells with the 3 combinations of oncogenes that will make our combination drug testing and nanoformulation drug testing specific for that rare disease. We have drug formulations that we have loaded with drugs that we know we have good loading and activity and good formulation characterization. We also have data showing how those combinations work in vivo in animal models with colorectal cancer. And so on every goal, we are making good progress with the intent that once we have the cell lines with the specific 3 mutations in goal 1, we'll be able to show you some of the results that you've seen already for the soluble formulations, but in that specific rare disease. And so as part of goal 1, we're going to expand and QC test the recently tested generated BRCA1, P53, BRAF V600E cell lines. We're going to continue to expand and quality control test the P53, BRAF V637 cell lines for the mouse model so that we have a mouse model that has this specific rare disease. And then we're going to look at, as part of goals 2, 3, and 4, to look at in vitro and in vivo activity of these soluble drug nanoformulations, including the PARP inhibitors, against this very specific rare colon cancer that has these 3 mutations that we've generated in goal 1. And then as part of goal 4, we hope to really identify the best drug combinations that we've seen from goals 2 and 3, and then see which of those work best when combined with immune therapy, and in particular anti-PD-1 in the CC26 model, so that we can provide guidance on what we think are good combinations to test for treatment of this disease. And so with that, I want to acknowledge and greatly appreciate the opportunity to do this research through the Gastrointestinal Research Foundation grant. We're very grateful to Katie and Leo for the ability to do this research and to advance the therapeutic strategies that we're investigating. And we look forward to any questions that are presented.
Thank you so much, Dr. Salem. Our next presentation is from Dr. Chris Mason from Weill Cornell Medicine. I first learned about Dr. Mason's work when he appeared on the Lex Friedman podcast. He works to better understand the tiniest details of each individual's disease so that a targeted and effective treatment can be developed and delivered. The results are providing an unprecedented resource for the study of human disease and biology, such as colon cancer. Please welcome Dr. Chris Mason.
Right. Can you hear me okay there? Right. I'm literally at an airport. I just gave a talk today at AGBT, which is a big genomics conference that's here in Florida. So I'm at the Ruby Tuesday, which so far seems to be good. So I'll pull out my slides here. And again, I want to thank you very much to the organizers for having me come by. I'm also trying to buy tickets back to the next meeting. Okay, pull this like this. So thanks so much to the Rifu Foundation. Thank you so much to Katie and also to Leo. I don't see here, but you certainly pass on my thanks. And really also to the committee for selecting me for this grant, which is that we're very grateful. And I'll walk you through a bit of what we're doing. That was, as you just heard, to break down really some fundamental questions about our knowledge of human anatomy and organs, particularly in colon and the GI tract, and also what it looks like in cancer. And I will try to keep this to 6 minutes and keep it also very high level. So every organ, as you can all imagine, has spatial organization. At some points, you can see these look like individual cells, but if you look hard enough, sometimes I think they look like stars as well. You can see the very beautiful structure. And oftentimes when we study disease or healthy cells, we study them kind of like this. We'll grind them up in bulk, or sometimes we can do single cell biology, but what we want to do to better understand normal and disease biology is through a spatial context, where it's the equivalent of being able to understand the platter on the right versus getting just a blended kind of mixture. You can see on the left was how a lot of biology and cancer is studied, but actually very much due to the benefit from the GI Research Foundation. This has enabled us to purchase a new machine called the Cosmics Spatial Molecular Imager, the SMI, which lets us enable not just RNA or protein, but actually both. You can see here we have other systems and technologies on campus, but this is the latest and the best cutting edge tool in spatial biology. And in particular, we actually published a review paper on this just last year, showing that what we have is now an ability with this machine, which is called the Cosmics, to get at the highest resolution, so down to 50 nanometer resolution inside cells, and also looking at thousands of targets at the same time. So it gives us, in the upper right corner is the greatest throughput with the highest resolution, and therefore giving us the best view of the biology as well as cancer when we look at cancer samples. So this tells you kind of our excitement around technology, what we can now see with it. So, and what we've been doing is building out, first starting a bit with some normal tissues and also adjacent normal, building out this, we're calling it a SAHA, which is the best acronym we could come up with in terms of what it is. It is basically the way to build out a map of normal anatomy first. And with that, we can get spatial expression, as you can see, tissue architecture, cellular interactions, and also then look at ligand receptor pairs, which I'll show you some of this data, which we're very, very excited about. And then also start to use this as a resource for the field. So all these data we've made publicly available will be used for many people. Actually, the meeting I just am coming from, people were very excited about these data and already requested to start using it, which is always nice to see. So this is a partnership, obviously, at Weill Cornell Medicine. We partnered with New York Genome Center, where I'm also a faculty, and also NVIDIA for a lot of the data processing. This is kind of a big AI challenge as well as there's a biophysical challenge. So this includes working closely with a pathology department at Weill Cornell, working with other collaborators here who work on anatomy, as well as on the technical side, NVIDIA and NanoString, and some collaborators also at St. Jude's. It's a very combined effort of many sites and many experts working together. And so far, where we're at is we generated 3.6 million cells across the body with a focus on the GI tract to start, especially because it's relevant for the GI Research Foundation. And you can see here we're doing the cosmics, we're doing high throughput, 1,000 plex of RNA, 64 plex of protein, and also doing whole transcriptome, which is 18,000 targets. Up here in bold is where it's filled in is what we've completed so far, and other samples are queued down the road. But we're focusing, as I mentioned, mostly in the GI tract and colon. And these are some of the images that are really quite striking, where we can start to see, of course, you can see the structure here of this tissue. But what's really extraordinary is you can also map, you can see here between individual cells, what are these ligand receptor pairs? And that's how cells communicate with each other, how they basically literally exchange information, talk to each other, regulate tissues, regulate homeostasis. And so these are things that we've been able to tease out here, you can see. But also you can see at the protein level up in this section. And if you look in the upper right, we can also see different proteins. So these are human leukocyte antigens, or HLA. These alleles help define the immune system function. And here we can see some of this heterogeneity within the tissue, and also likely what this immune system, what these cells are doing as they communicate, in this case, in normal tissue. But then we've also begun to put together basically across the entire GI tract. You can see on the left, all these are different kinds of cell types. So this is stromal cells. You can see B cells, T cells that appear across the GI tract. You can also see there's some images of zoom ins from the appendix, the ileum we've looked at. You can see it's a lot of colors because it's many, many cells. It's millions of cells that we can map onto one reference. And you can see, even though you can have the same epithelial cells, for example, the appendix, you can see here some of the structure. Whereas the ileum, you can see they're, of course, used differently in different tissues along the GI tract. And so this is what we've been kind of seeing from some of our data so far. Again, quite beautiful images. You see this high resolution individual cell and then intercellular communication. And then this is, for the most part, these ligand receptor pairs are often correlated between each other, but some of them are, the highest ranking ones are different. So you can see what's at the top of the appendix, which is the ileum, for example. These ones are distinct. And you can see some of the labels here. So these very, very specific and quantified interactions between the cells. But the thing I want to close with is what we've seen from the colorectal cancer from some of the first samples we've processed. And again, this is the same kind of imaging data, but now you can see in these particular interactions, normally in a healthy colon, you'll see that things are along this dotted line and they're the same as in cancer. But if it deviates like this, you can see all these different interactions here are different. They're enriched in the colorectal cancer sample, meaning this is a different feature of the biology that we're learning about with this new spatial imaging method, and getting frankly, the highest ever resolution of a colorectal cancer or of any of these tissues. So it's really striking to see some of these differences. And then when we annotate them with what's in the literature, we can see that some of them are known. This one is driving metastasis potentially, and CCAM1 can control proliferation. But these other dots on here are entirely novel discoveries. This gives us a new discovery, new targets. These little dots here tell us new kind of interactions in the cancer that we don't normally see in normal tissues, which gives us new targets, new understanding, and I think gives a lot of excitement for what we can discover in the upcoming tissues. And so what we're doing next is basically using these data with also a partnership with NVIDIA, who I just met with when I was here at this meeting, where they really want to use, they're calling it spatial GPT as a way to actually build some of these new AI methods to find new features in the cancer samples and to better understand some of the biological changes. This is a lot of work on what we're doing now. And I like to think that this technology is going to take what used to be, 19th century histopathology, and of course, we're all used to molecular pathology these days, but I think now and in the future, we will get more towards a digital pathology and have a real in-depth and integrated multimodal and multi-omic view of what's happening in these tumors, and then better use that to diagnose, and then also guide treatment for future patients. And then ideally, instead of going one treatment for all, we go to more personalized diagnosis over time. And I wanted to keep it about 6 minutes. So that's I really cannot be here without having an amazing lab. I'm very, very blessed to have extraordinary people I get to work with that are fun and inspiring and work very hard, and even work on the weekends when I tell them not to, but they're really wonderful and work all the time. And of course, thanks to the other lab members, other collaborators. And finally, thank you so much to the GI Research Foundation, Katie, Leo, and all the directors. And thank you so much. I've been taking questions.
Thank you, Dr. Mason. Tonight, Dr. Arturo Ramirez is with us from RareSight. Dr. Ramirez has researched cancer diagnostics for 2 decades. Dr. Ramirez and Dr. Pashtun Kasi will discuss their work to improve early detection of cancer through advanced analysis of circulating tumor cells, or CTCs. A deeper understanding of the makeup of CTCs allows doctors to select therapies to specifically treat a person's cancer and to determine in real time if the patient's cancer is responding to the drug treatment. Please join me in welcoming Dr. Ramirez and Dr. Kasi.
Thank you very much for the introduction, Katie. Let me just share my slides. Okay. Yeah, thank you very much for the introduction, and thank you so much for the support from GIRF and Katie and Leo. It's very appreciated. Thank you so much for also this opportunity to present this work tonight that we've been performing around the comprehensive precision biology testing to enhance colorectal cancer patient care. So RareSight, as a precision biology company, our goal is to provide clinically actionable data from cancer patients to the physicians so that patients can receive the best possible treatment for their particular tumor. Now, we provide testing of blood samples as a liquid biopsy of the tumor. And why is blood such a good source of information? Well, blood can be readily obtained. It provides a complete understanding of the tumor, and it contains circulating tumor cells, or what I'll call CTCs. And these CTCs are prognostic and predictive of disease. Because the CTCs originate from inside the tumor, they have the same characteristics of the tumor, and they can be used as a window into how the tumor evolves over time with minimal risk to the patient. Now, besides the liquid biopsy precision biology services we offer, RareSight also generates highly multiplex spatial biology testing of tumor samples with our Orion platform. I actually just saw on one of Christopher's slide reference there to Orion. Nice to see that. Yeah, thank you. So as you can see, here is our Orion platform. It's very similar to what he was mentioning, where Dr. Kasi actually and his collaborators recently published a Nature Oncogene manuscript describing the exceptional response to a novel immune oncology therapy called BOT/BAL, and you can really see what's happening at the tumor level with this technology. So as an update to the liquid biopsy analysis that we're doing within our GIRF-sponsored program with Dr. Kasi and his patients, we performed testing on 44 different patient samples, analyzed within those 44 patients 156 blood tubes, and have found CTCs in 95% of patients with an average of 59 CTCs per blood tube. Moreover, the CTC number and how it changes over time for each patient correlates with the treatment response. Also, CTC biomarker expression shows a very strong correlation with tumor tissue. And importantly, CTC testing can be performed as soon as a few days after initiating therapy, so we can obtain real-time results on the patient's response to therapy. So as a sneak peek into what innovations are coming down the pike for RareSight, what we're proposing next is a complete paradigm shift in liquid biopsy testing, and we're calling it the multi-analyte liquid biopsy, where in addition to the CTC enumeration and 2 biomarker testing that we currently provide, We would also measure tumor-derived extracellular vesicles, single CTC next-gen sequencing, cell-free DNA sequencing, and a complete quantitative characterization of the immune system of each patient from one tube of blood. So such a comprehensive view of a blood sample has never been done from a single tube of blood, and would constitute a monumental increase to the amount of data that's available to a physician for their patients so that they can make better informed treatment decisions. We're very excited about this work that's coming up. So with that as an introduction, I'd like to pass the microphone over to Dr. Kasi, and he's really been instrumental in all of these studies. He's been a close collaborator to Raricyte since 2020, and he's studying colorectal cancer using these liquid biopsies. So Dr. Kasi is the Director of Colon Cancer and Precision Medicine Research for Liquid Biopsies at Weill Cornell Medicine, and has provided all the patient samples for testing. So with that, Dr. Kasi, the floor is yours, and I'll be clicking for you.
Well, thank you so much for the kind invitation, and nice to see some familiar faces. Building upon what Chris was talking about in terms of knowing about all the cells in multiomics, and also what Arturo presented. A lot of the folks on the call are familiar with liquid biopsies, and a lot of these are getting a lot of traction in the clinic. But the broad umbrella of liquid biopsies, when the people talk about liquid biopsy, everybody's talking about circulating tumor DNA, or ctDNA for short. Now, while that gives us a lot of information, it is not necessarily all the information that potentially could be obtained, especially in a tissue sample, in terms of the actual cell surface proteins and stuff. And there's always going to be value for multiomics and actual staining and understanding the spatial heterogeneity that Chris showed. I guess one of the challenges over the last 4 or 5 years, we've been trying to work with Raricyte and trying to show the feasibility, because to date, at least for colorectal cancer, the general assumption is you cannot capture cells in the bloodstream, or if you had to, it's only in select academic centers. It has to be the same day or a few hours, and protocols that are not often reproducible. So the data is all over the place in terms of the feasibility. And the question would come often is, why do we need cells? I think the cells, with the intact cells, has a lot of downstream potential. Again, not meant to replace the DNA testing, but in terms of supplementing it. Similar to like liquid biopsy is not meant to replace tissue testing. CTCs are not meant to replace ctDNA, but can help complement it, because you can see the actual cell surface protein. We've developed some new markers as well that could be used for guiding treatment. These cancers also change and evolve over time, so you can obtain all this information from an actual intact cells, and potentially you can even grow them, from just a simple blood draw. So some of the work here that's summarized here, for example, just the basic question of, is it even possible to detect CTCs? One practical thing I want to mention is, Raricyte is in Seattle, we are in New York. One of the things that Raricyte has done, which is ahead of its curve of other platforms that are out there, is as opposed to other CTC platforms that need processing within a day or two, or within X number of hours, the tube of blood that we're able to ship to Raricyte is good for almost 9 days, which allows, I would say, decentralizes the analyses. So it's not something that's only available in the future for patients that I see, but to anybody and everybody that can get a blood sample for the analysis. So shown here in these curves are all these patients that have not just a few cells, but dozens of cells, especially for colorectal cancer. They tend to be more shedders of cells in DNA. If we go to the next slide, we also showed the value of doing testing like HER2. Like in this particular case, you can see how broadly positive the HER2 biomarker is. It's a target for which there are dozens of drugs now, and it's going to be probably the first marker in the next few months that will get agnostic approval, meaning doesn't matter what type of cancer you have, as long as your cancer is HER2 positive, you can get a drug for it. So knowing that is somebody a candidate for a therapy that works really well, we published a report of a patient whose cells just disappeared within days of starting therapy. So again, traditionally as an oncologist in clinic and as patients and caregivers, we are looking at scans and tests every 2 to 3 months. here with CTCs, with Raresight, we're able to know within 48 to 72 hours if something is working or not working. You can see these case examples, of which we've published many, where these dozen CTCs literally disappear from the bloodstream way before anything happens in the bloodstream. Of course, we see notice in the patient's improvement. That's always a good sign, but it's good to tangibly see the improvement with these quick turnaround. And turnaround is within days. We have a weekly rolling call. We literally get the results back within a few business days of getting the samples to their site. Next slide. And again, just wanted to kind of highlight some additional examples. Again, on scans, you see the improvement from left to right in a patient, young onset colon cancer, for which there is unfortunately an epidemic that's going on. But then CTCs could go down as early as within days of therapy, as well as tumor markers and scans take months. We'll go to the next slide as well. Again, just as they say, picture is worth 1,000 words. These are additional examples of things changing very quickly in the bloodstream of a patient, and also capturing changes over time. So that, again, I want to be thankful for support that GI Research Foundation has provided. I would also mention it's not just support. I think GI Research Foundation and the work that Katie and the board and Leo have been supporting, I think it's also been a huge collaborative network of recognizing all these other scientists that we are collaborating and actively working with towards hopefully a cure for diseases like colorectal cancer. So again, thanks so much for the kind invitation and the opportunity.
Thank you, Dr. Ramirez and Dr. Kasi and Raresight. I think now I'll turn it over to Jackie, our executive director, who will share with the group any questions that may have come in.
Thank you, Katie. So Dr. Taraga had to go into surgery. He believes he's going to be able to join us shortly. So we're going to pause for questions. And we've got a few that have come in. So mostly what people seem to want to know is how long, if everything goes as you're predicting, how long do your treatments or your new therapies or new ways of diagnosing or new understandings, how long before they're actually in the clinic and becoming the standard of care for how we address cancer? So Katie, I'm going to let you start, because we did get a chance to ask Dr. Salem that question before he had to leave, and he shared something with you that you could share with the group.
Thank you, Jackie. So I think when Dr. Salem was asked the question about the nano delivery mechanism of drugs that are already available but would be better served in the special way that he'd like to do that, the answer was about 1 to 2 years.
Great. Thank you for sharing that.
And maybe if I can take a stab at the question, I would say it's not necessarily how much time do we need. I think some of this is already in clinic. Some of the things that we just heard or talked about directly or indirectly are propelling our understanding of new drugs and trials. Arturo mentioned, a new immunotherapy trial that we just presented a few weeks ago that potentially might bring a new immunotherapy option for patients with colon cancer. Hopefully it will get approved, I would say, sometime this year, and already patients are getting it through compassionate use. But it was, analyses by Raresight, similar to like the platform Chris was talking about, that gave us a deeper understanding of the mechanism and allowed us to, do robust analyses, even in just a handful of patients, to really help inform care. Whereas traditionally you would have needed hundreds of patients, years of follow-up, and analyses that sometimes happen, decades later to know or to move the needle. I think with all these things happening in real time, we've been able to, I would say, accelerate the progress of delivery of these things in clinic already as we speak.
I'm going to go, Dr. Mason, go ahead, and because someone specifically asked, what are the barriers to, what might be the barriers to someone being able to have their disease looked at in the way that you described as a regular part of healthcare?
Yeah, I think so. We'll get there eventually. But right now, the biggest factor is cost. Actually, so a lot of those images I showed you, they're beautiful, but they're expensive. They're like $4,000 per slide, and so and that's before you have anyone look at the data and process the sample. So today it's expensive, but the hope would be that in the coming, I think that's one of those cases where to get it as cheap as histology is, where you see those kind of pink and red slides, where those are really very cheap, and $20-$30 to get down there will take some more time and automation and innovation. But I think that's still in a matter of years, not decades away. So because a lot of the technology is very new, so it's the first version, it's the lowest throughput variation. But we're working closely with some of the manufacturers, working with the pathologists, and the more we can make it automated and routine, I'm hoping that right now, if someone wants it done, we're happy to do it. It's just a matter of cost, but eventually it'll be routine, hopefully in a matter of a few years.
Dr. Rubin, you have your hand up, but Dr. Taraga is here. Can we let him go first and then have you do your question after?
I'll just thank everyone. I can ask my question at any time. And of course, it's nice to see you, Karen. So I'll let you go.
I was lucky enough to meet Dr. Taraga at the University of Chicago before he left to join Yale Medical Center. Dr. Taraga continues to collaborate with scientists at U Chicago Medicine to advance a novel technology called liquid biopsy that measures the DNA released by cancer cells for earliest possible detection. Please welcome Dr. Kiran Taraga.
Thanks, Katie. That's it's so nice to see everyone. I have to apologize. My I was in the operating room actually and just finished, so it was perfect timing, and that's why thank you for adjusting the time. So I think I thought I'll just give an interim update a little bit. So first of all, thank you so much to GURF and Katie and CACure for helping us with this grant. I think it's remarkable what partnerships between patients and physicians and researchers can actually do when we think about advancing the field. So I think this is very important, and so thank you very much. I don't have any disclosures about this, but I wanted to frame my update a little bit more broad-based and generic, and then I'll get into specifics depending on how much we want to get into it. But the challenge that I do, and in fact, what I just did today as well, is a patient, who had peritoneal metastases that was just not visualized on scans. And this is something that we face on a daily basis. And when you really think about what the peritoneum is the sac that encloses all the organs of the abdomen. So you can see that when you look at this sort of graphic and you see patients who have a liver and your stomach and intestines and colon, you can actually see the dimension that they have. And this sort of peritoneum, which is the sac that is encasing all of this, is actually a very thin sac. It's like saran wrap. And the problem is when you get a scan, when you get any type of imaging, it's very difficult to discern tumors on this, even though you may have numerous tumors on the peritoneum itself. And so I think this is a big problem from just a logistic standpoint, an imaging standpoint, and how you actually look at this. In fact, so much so that, in 2016, some scientists believed that the peritoneum and the mesentery, which, all of you that are interested in the field of colon cancer can sort of see this 3D model, which basically looks at the colon, and the blood supply of the colon is called the mesentery. And the mesentery is invested with the 2 layers of the peritoneum. So big challenge for us as doctors, as surgeons, when we're taking care of cancers to these areas. And it was actually called a new organ because of its unique immunological phenomenon and its unique way of acting. And so if you really think about colorectal cancer and peritoneal metastases, there's estimated 13,500 to 27,000 new patients with peritoneal metastases. So think of it, put it in frame of reference. There's 35,000 new pancreas cancer cases every year. You know, the number of clinical trials that are enrolling patients on peritoneal metastases only in the United States, it's essentially close to zero, because you just can't enroll these patients on trials because they have undetectable disease. It's very difficult to bring them on. And in fact, even till today, if you look at the NCCN guidelines, which is a body that kind of defines guidelines for patients with peritoneal metastases, you know, often you say, well, just take care of symptoms. So this is a group of patients that's just kind of relegated. You know, it's very difficult for us to put them on trials. It's very difficult. Many surgeons across the country don't know how to take care of this. And so it creates a big problem. The second statistic I want you to all to kind of take home for sure is that 20 to 40% of patients with T4 colon cancers will develop peritoneal metastases, which are occult. So almost a fifth to a half of patients with T4 colon cancers, and this has been seen in clinical trials. And the worst part, and I know you've heard some lovely talks, and Dr. Kasi is here, so I always feel a little embarrassed talking about circulating DNA in this matter, but what we have also seen is that ctDNA, so circulating tumor DNA, is actually is present or detectable less than 50% of the times when patients have peritoneal metastases. In fact, we just published a paper where when patients had peritoneal metastases, only 25% of our patients had Signatera at a spot check. And it's only when you did it in a recurrent multiple way fashion that you can actually identify those patients. And so it's a big deal as compared to, say, when you have a liver met or lymph node met or lung mets, when you can quickly see, you know, ctDNA rising. And one of the big problems we have with that is that when we're actually getting the DNA out of the blood, we use a technology called bisulfite DNA extraction. So it actually destroys some amount of DNA, and you need either large amount of DNA in the blood, which is being shed by this tumor, that then you can pick it up and do it. And the peritoneum is this barrier that actually keeps the cancer within it and doesn't let the DNA shed out. And so the amount of DNA in the blood is usually very, very low. And so our conventional technologies fail us. And certainly a lot, you know, as our technologies are getting better, we're hoping we'll have a better way of doing this. And so I was fortunate to work with Xuan He, who's one of the chemists at University of Chicago, who's probably going to win the Nobel Prize at some point. But what Xuan's lab has discovered is essentially using a nanoseal. So it's essentially a biotin molecule that binds to the DNA and actually allows for extraction of DNA at very, very low concentrations. So when we can pick up as much as 1 nanogram of DNA in a plasma from one tube of about 5 ml. You know, this is sort of like our future goal would be some point getting to like pinpricks to identifying you know, tumors in the blood. And this has been validated. It can actually find polyps from cancers from metastatic cancers. But, you know, our question was really, can we use this same technology to get to the next level where we can not only find peritoneal metastases, but also maybe understand more about them? And so this is the work we've done so far. We've already done this work in about 65 patients. And so thank you to GERF for getting us to do all of this work. Our plan was to look at this hypermethylation signatures. And so for those of you that are not very scientifically inclined, I'll kind of tell you what this graph is saying. What we found was not only that can we make a signature that is specific to patients with colon cancer peritoneal metastases and identify them at very high sensitivity and specificity rates, we can also make it in a cheap way, we think. We can identify, so the first 3 genes that we have, we can use that to build a sort of very easily reproducible technology with which we can identify the DNA in the plasma. But I think what I'll bring your attention to is this IL-10. You know, we've also uncovered this, and in fact, fortunately, one of our other colleagues at University of Chicago has just published another paper looking at the immune profiles that are changing for peritoneal metastases. And we're certainly very interested in this premise that we're seeing specifically for peritoneal metastases. And in fact, what we've also found is one of these genes that is hypermethylated in the signature that we found is actually also associated with survival. And so actually, and has druggable targets. So it does have druggable targets. And so, you know, I think this is work that we are hoping to validate, which is, you know, our specific aim 2 for the grant. And we're hoping that, you know, over this coming year, we'll be able to get to doing some of that work. looking at it in the tissue as well and confirming that. So we're right now in the process of going through some of the logistics of getting that work going. But I think certainly very interesting in terms of where we are. I think another thing that the grant has given us, which I think is very helpful, is that we are hoping to build now through EPIC, which is one of our electronic medical records, a national registry that will then also link biospecimens and imaging, and essentially then facilitate and accelerate the work of, say, radiomics, or bringing the group of us that do this work together, the peritoneal metastases work, to kind of get together and start thinking a little bit harder and accelerating these discoveries, more. I think the work has also supported the salary of one of our research fellows that has actually helped with building something called the PSM Consortium, where we now have new guidelines. You'll hopefully see in the next few months some of these new guidelines coming out for the management of these patients. For the first time, we've worked with NCCN and ASCO and SSO and all the colorectal surgical societies, patient support groups, including Colon Town, to try to get the management of these patients changed. And I think what is very exciting since I came to Yale, and this was not part of my grant, but I think certainly very exciting and interesting, is one of my scientists, Jason Schelzer, has done some remarkable work in something called chromosomal engineering, and his paper was just published in Science. But the basic premise is that we have known for about 80 years that all cancers have more chromosomes inside their cells than non-cancer cells. This has been known for a very long time. And for those of you that are wondering what are the more chromosomes or less chromosomes, it's sort of like Down syndrome, or you might have heard of things like Klinefelter syndrome. These diseases, these genetic diseases that happen because someone has an extra or a less chromosome. We know that cancer cells dominantly have multiple copies of chromosomes. And we always work on mutations. You saw my previous work was also looking at mutations. Can I alter the BRAF mutation? Can I alter the BRCA mutation? Can I do this? Can I do that? But the concept is, can you actually change the whole chromosome? Can you actually change the entire locus on a chromosome? So can you change a certain segment of the chromosome called chromosomal engineering? And essentially what we found, or what he has found, I shouldn't take credit for that work, but what he has found is that you can actually engineer these chromosomes. And there's no question colon cancers have very significant aneuploidy, which maps to chromosome 1 as well as chromosome 4. So there's these two chromosomal aneuploidies that he has discovered and is working on. And so I think just with the work that we are doing currently, our hope is to then use that to understand aneuploidy in patients with peritoneal metastases as well. So I think it's an exciting new area that we're taking forward. So thank you all for listening, and thank you to CACure and GERF for this super grant that has helped us accelerate our work. So thanks.
Thank you so much, Dr. Turaga. I will hand it back to Jackie, who was going to field the remaining questions.
Okay, Dr. Rubin, you're up.
First, let me thank our scientists, and I'm sorry Chris had to run to catch his flight, but I really appreciate the work you're doing. I also appreciate you updating our board members and the others who have joined us on this call and who will see this recording. My question is a little bit about the heterogeneity, or the different types of tumor behaviors that all can coexist in a single patient. So what we've heard examples of in this incredible work that's going on is identifying very specific targets and understanding the unique cell signatures that are characterizing a tumor. And while some cancers, as the scientists certainly know, are clonal and look that way, I certainly appreciate that they often will change over time, or that they have behaviors that represent multiple different pathways. And so I'd like in lay-friendly responses just for you to help us understand the dynamic features of tumors as you treat them, and how you have thought about this with your different approaches. And maybe we could start with Arturo or Pashtun, Dr. Ramirez or Dr. Kasi, and then we'll hand it over to Dr. Turaga.
Yeah, no, I think very rightly put. I think there's changes, these cancer cells in different parts of the body even in the same person, are not necessarily just one disease process. You know, to the eyes of a CT scan or an MRI or a PET CT, these may look like spots, but as Dr. Krager also showed, these are different organs, and we're also learning that even the same drug in the same person having the same cancer in different parts of the body, we're seeing not necessarily the same results. You know, the liver seems to be a separate battlefield. I tell sometimes patients that's like having children. You know, even if you have identical twins growing up in the same household, they don't necessarily have to have the same personality. So I think we also have to be creative and innovative about how to address. And as I was hearing some of these talks, I think it also kind of shows the complexity of dealing with these challenges of cancers in the different parts of the body. How are we viewing the liver as a separate challenge, as how surgeons like Dr. Raga are helping address the peritoneal in a totally different way, and how work like with rare site with circulating tumor cells is kind of capturing the heterogeneity or changes over time in the same person or from the different parts of the body with these novel liquid biopsies and stuff. I think but it's good to also see that I think increments are happening in these silos, but overall I think they're helping move the field forward for that each patient, because we would end up using more than one strategy here.
Yeah, I guess I would second everything that was just mentioned. I think you brought up a very important point about heterogeneity within a tumor. There's heterogeneity also with different metastasis in the same patient, and then moreover, it just changes so much over time. So both with the cell-free DNA that Dr. Raga was talking about, or the CTCs, you at least can gauge how that happens over time. And we really want to test and treat a patient with the tumor they have today, not with the tumor they had 2 years ago before different treatments that comes from the tumor sample. So very good question. I think all these technologies are helping to just give more complete information on what's happening and in real time.
I mean, I think, as a surgeon, one of the things we face is every day when I'm operating on patients, David, I mean, not only the genomic heterogeneity, there's even heterogeneity of how they present, where they're located, what they look like. I mean, it is just remarkable that these diseases are incredibly heterogeneous. I think the other thing that I also struggle with is, this is not just cancer in isolation, right? We're a system. So I think it's always the question of the host, the immune system. How do you kind of actually interplay, and how much does heterogeneity matter? Like, where does it matter? And I think that piece is fascinating. We know that peritoneal metastases are often the CMS4 type colon cancer. So they kind of fall in this broad group of cancers, which are a little bit distinct than the, other types of colon cancers. But, even then, they're still not, homogeneous enough. So even the group of peritoneal metastases, or even if you took mucinous peritoneal metastases, they're not homogeneous enough that I could say, this is how we treat all of them. But I think on the flip side, without being so nihilistic or being like, oh my God, it's so heterogeneous, I do think that if we can understand the immune or at least the host response to a lot of these, many times, removing all the clones may not be necessary. Maybe we can work through, removing a certain dominant number of clones, or, some way of approaching them where it's, which is why we cure cancer still, right? I mean, we still cure even metastatic cancer in many patients. So it's kind of a remarkable thing.
So thank you, everyone. We're at our time, and if we have other questions in the Q&A, we will be asking our investigators to provide short written answers to any remaining questions that we can send out to our participants. So thank you so much. And I'll turn it back over to Katie.
So thank you so much to all our presenters tonight. We're in awe by your innovative thinking and the progress that you've made. A special thank you to Dr. Rubin for your support and your leadership at the GI Research Foundation. Your commitment to our vision has made this initiative a smashing success. And thank you to our participants. So we're very glad that you could join us and be part of this learning journey. Our donors and constituents are the fuel behind the funding of our research, and we couldn't do it without your support. We have our next session with the few other projects that we funded on March 20th, 2024 at 5 p.m. And just one more time, thank you and have a great rest of your evening. And thank you for sharing our mission to transform lives through science. Good night.
Thank you.
Bye.